Today I'm going to be talking about machine vision phenotyping uncovers novel relationships between kernel and seedling characteristics. And this has been a really nice collaboration between us and a group from the University of Florida, where I'm currently a postdoc in Mark Settle's lab, and the University of Wisconsin. And there's several PIs at Wisconsin, Edgar Spalding, Nicola Ferrier, who's in the engineering department there, and Nathan Miller, who's in Edgar Spalding's lab. And it's been a very nice collaboration to do a lot of this work. And I'm presenting a lot of work here that I did not do personally. The very first part of the talk was mostly me, but then a lot of the work towards the end is a collaboration and has been done by other folks, and I'll acknowledge them as we go. So I start with this very simple schematic as a way of representing the idea that we have a lot more tools for studying genomes than we do for studying phenotypes. And so an example of this, you can open up any mapping paper recently, and you'll see that we have thousands, if not tens of thousands, of markers against what probably is usually only a handful of phenotypes. And there's a reason for this. Obviously, phenotyping is expensive. It's hard in some cases to do. It's laborious and expensive. And so what we're trying to do, and so this sets up the issue of, I don't think we're quite in a bad situation as this donkey is in, where I don't think he's going to make any progress at all with this cart. But it's analogous in that in both these cases, I think what we're lacking is a lack of horsepower. And so you can imagine if they strapped a Clydesdale to this cart, you'd be moving along just fine. And so I think what this illustrates is I think we can make progress faster in making discoveries if we add a little horsepower to our phenotyping capacity. And so that's what my talk is about today. Is we are going to be talking about relating kernel traits in maize to both within different kernel traits and to seedling root growth traits. And this also serves as a good overview of my talk. I'm going to be talking mostly about the very first two components here, which are the weight and the NIR spectrum, which is what I do. And then in the second part of my talk, I'm going to be talking more about digital imaging, kernel morphology, and then time-lapse imaging of seedling root growth. What's important to know here is that each seed, so the first three technologies I'm going to be talking about are non-destructive assays. And so what that allows us to do is to give each seed a number and track that one individual seed through each one of these platforms, and allows us to get what is equivalent to a longitudinal study of every seed through the pipeline. And that really gives us much greater statistical power to detect associations. And I'm going to be talking about that statistical power briefly later in the talk. And I think it's really giving us a much better insight into the relationships that exist between these traits. So as I said, the part of the work that's being done in Florida is mostly with NIR and kernel weight. And so I'm going to be going through that in much greater depth than the other components. So what is NIR? So NIR stands for Near Infrared Reflectance Spectroscopy. And why is this useful to us? Well, it turns out that organic molecules absorb light. And so not only near-infrared light, but they also absorb infrared light, which is a much lower wavelength, so 200,000 to 500,000 nanometers. And so why do we use NIR instead of infrared light? So infrared light is very good at detecting. So organic molecules such as the oxygen-hydrogen, the nitrogen-hydrogen bonds that I'm showing here absorb infrared light very strongly. And so in the infrared portion of the spectrum, you get these very strong peaks, which are very specific to those organic molecules. However, these organic molecules absorb that light very strongly. And so what that means is that if you have a sample that's very thick, the light gets absorbed within the first few microns and doesn't pass through the sample. And so therefore, it's not very effective for us to be able to use to measuring thick samples such as seeds. Well, this is why near infrared is so useful, because it's what we consider a secondary absorption. So you're what they call overtone. You're absorbing what are called overtone wavelengths, and so but they absorb them at a much weaker. The absorption is much weaker, and so what that allows it to do is that the light does penetrate through thicker samples. And so we can use NIR light on much thicker samples, as I said. It uses wavelengths between 800 and 2500 nanometers. Organic molecules such as protein, starch, and oil absorb water in this region. Reflected light is correlated to the composition of protein, starch, and oil. And what's really nice about NIR is it requires minimal sample preparation. So you can basically take a single sample, shine NIR light on it, and you can get your measurements from that. So NIR is a standard method for determining bulk kernel composition. I'm showing you here two different commercial machines that are used to collect bulk NIR. So these machines, sorry. As I said, these machines collect bulk NIR, so you would take several grams of kernels and put them in the machine, and they're going to give you a measurement of protein, oil, starch, whatever they're calibrated to detect. But we wanted to scale down this technology to allow you to measure single kernels, which allows you to do much better, allows you to do genetic studies on segregating material. And so this is a schematic of our single kernel NIR machine. And so the process for measuring a single kernel is, the first is the kernel is put onto falls onto a microbalance where a weight is collected. A puff of air pushes that kernel down an illuminated light tube. That tube is illuminated by 50 halogen lamps, which are these little sun-like objects up here. And so the NIR profile, the light reflectance from this seed then gets carried by the fiber optics that are sitting at either ends of the glass tube to an NIR machine where the NIR profile is collected. And as the seed falls through the glass tube, it can fall into a microtiter plate so that we can keep track of each seed as it goes through the machine. So this is a picture of the NIR machine as it sits in our lab. And then this video is a video of our lab technician, John Beyer, using the machine. I'm going to show it to you here. It takes about 6 seconds for every kernel to fall through. And in that 6 seconds, the microbalance collects a seed weight to within 1 microgram. It collects an NIR spectrum from 907 to 1689 nanometers at 1 nanometer interval. So you're getting 790 different traits, wavelengths for each kernel that falls through. And as I said, the seeds can be indexed into microtiter plates so they're all kept organized. So this is a typical NIR absorbance profile that you would get from a seed. And as you can see, there's no prominent peaks here that would be corresponding to a particular organic compound. And so what this means is that any one individual wavelength is not very accurate in being able to predict a particular trait. And so what we do instead is we use multivariate regression on the entire profile to try to estimate traits from individual seeds. So I'm going to walk you through the process of how we develop the calibrations for prediction. So the first thing we do, obviously, is collect an NIR from an individual seed. So then those seeds are then ground up and sent off to whatever analytical technique that you want to measure. So for example, if we want to measure starch content, we would grind them up and then submit them for starch measurements. So with those two things, the NIR spectra and the analytical measurement, you develop a calibration model. And why I have 66% here is because we do this, 66% of the kernels are used for calibration of the model. And in order to calibrate the model, there's a variety of different techniques that you can use. If you guys are familiar with genomic selection, you know that there's a myriad of different techniques for associating highly multilinear traits such as markers to a particular phenotype, right? And so the way we use it is partial least squares regression. We were talking this at the lunch about using other methods such as Bayesian methods or machine learning methods. And to tell you the truth, we've tried some of those methods. We tried ridge regression methods, deflation methods, partial least squares regression, which is a simple, relatively old method of doing this. One of the original ones that was used for doing calibrations is still the best. I mean, some of the other methods, such as Bayes A or Bayes B, come close to doing the accuracy of partial least squares regression, but partial PLS seems to work best in our hands, in our experience. So once you have the calibration, you can then take a novel spectra. So you've developed your calibration with 66% of the kernels. You can then take a novel spectrum and, using the calibration, get a prediction of whatever composition that you're looking for. And so then, since you have the analytical data for the remaining 33%, you then compare the predicted value to the actual analytical value to do your validation. Does that make sense? So we've done this for a variety of different components of the seed. And here I'm showing 3 different ones from a relatively old publication that we use. So this is % starch, % protein, % oil. On the x-axis is the predicted, is the actual amount of the component, and on the y-axis is the NIR predicted value. So as it falls along the one-to-one line is how accurate we are in estimating each one of these components, along with the R² value. And so you can see that the NIR does fairly well at predicting starch, protein, and oil. And I want to also say that we use, for those familiar with PLS, we only use 6 or 7 for these traits. We only use 6 or 7 PLS factors, and we don't drop any data, which is, I think, unlike a lot of other people that are doing PLS, they tend to drop things that look like this because they believe that those are erroneous analytical values, right? Instead of actually incorporating all of the data. So how does our single glass tube NIR stack up against other single kernel NIRs or against the bulk grain analyzers? And so on the first line is our accuracy for different components for the glass tube that I just showed you. And then other single kernel NIR techniques have similar but a little bit higher error rates. And then you'll notice that the bulk grain analysis devices have at least 1.5 to 3.5 fold reduced error rate as compared to our single kernel NIR. So those bulk devices are actually more accurate. However, given the fact that we're adding the advantage of having a single kernel prediction, we think that having this error rate is perfectly reasonable. So I wanted to go through one specific example that we recently used the single kernel NIR to calibrate on, and that's single kernel density. So density can be measured by using a CT machine. This is similar to a medical CT machine where you pass a human through, but this is a micro CT, so it takes much smaller samples, the size of a single kernel, for example. And so when you do one of these micro CT measurements, you get this is what a single slice of the kernel looks like. And the whiter the pixel on here, the higher the density, right? And so what you can tell very quickly is that the embryo is, particularly the scutellum, is much higher density than the endosperm of the seed. So what you can do once you have all of the micro CT is going to create slices along the seed, and then you're going to submit it for reconstruction. And what this is not an image of the seed. It's the 3D reconstruction of the seed based on the density measurements from the micro CT. And so what we can get from this are multiple measurements. Primarily the density of the entire seed and the volume of the entire seed. I have total and material on here. The difference between these two caveats of density is simply airspace. So total includes all material and airspace that could be inside of the seed. And then so what that allows us to do then is to also get a measurement of air inside the seeds, or void space inside the seed. So I'm showing here an example of a known soft endosperm seed, opaque 2. Opaque 2 is a protein quality mutant, so it has protein accumulation defects, which primarily makes it accumulate soft, starchy endosperm, as compared to a normal seed, which has this layer of vitreousness around it, which is a much more dense starch. And so you can see, if we set the threshold properly, that the micro CT can very easily distinguish between the normal kernel and the soft endosperm of the of the opaque mutant. And so similar to the oil and starch, we can do a calibration and show, so we ran approximately 350 kernels through the micro CT machine. It took about 3 months to do, an entire summer for an undergrad. And what you can see here, if we do PLS regression, is that the actual measured material density and the predicted material density line up quite well. And so we're able to say that we can actually estimate material density and total density and material volume from the NIR quite accurately. So this gave us a new phenotype to use the NIR for. And what's nice about the NIR is since we save all of the data, we can back project this calibration onto all previously collected data to extract density from all seeds that have ever passed through the machine. And so it's actually quite powerful. Developing these new methods for the NIR turned out to be quite powerful. One of the nice things that turned out from this project was that our single kernel density measurements actually correlated quite well with test weight. So test weight is a measure of yield, as a metric that you judge hybrid production by, and it's measured in bushels, pounds per bushel, which is a measure of density, right? And so what we did was we requested lines. Jody Edwards sent us lines from Iowa State, where he had 30 hybrid lines, and he had field trial test weights for each one of those lines. And you can see that our single kernel density prediction lined up quite well with his field measured test weight. And so what this gives you an ability to do is to use the NIR machine to possibly increase test weight of hybrid lines by a very simple 4-second assay. You drop it through the machine, and then you can tell if it's going to have estimate if it has a high test weight or a low test weight. Okay, so that was my introduction for NIR. And I think I showed you that the NIR can detect composition and nutritional quality traits. We also recently found that the NIR can see density quite well. The signal's quite good, and that you can use NIR to potentially estimate yield parameters such as seed size and weight and test weight. So now I'm going to move on to, so I've been speaking about the intrinsic properties of the seed, meaning that how much oil content or protein content is within the seed. But we can also we think we can extrapolate NIR to more extrinsic factors, such as the environment that it came from on the ear. So kind of like reversing the correlation back to the environment that it developed in. Or it might also be possible to use NIR to predict how a seed might perform if you put it in the soil and let it grow. Right? It's not a radical thought to think that the composition of the seed might have an influence over how the seedling grows when you germinate it. And so these are more extrinsic factors that we think we might be able to use NIR might be used to estimate, or to have be able to say something about those parameters just by sending it through the NIR. So the first thing I'm going to talk about is kernel morphology. And so what we do is after the seeds are sent through the NIR machine, they're sent to Wisconsin, where they are placed in front of 3 fixed position cameras, where we get a side, an abgerminal, and a cap image of each kernel. And so these are still in indexed order, so we can associate all of the data. And then we also get a cap color area. And so there's a variety of metrics that you can pull from those images, but today I'm just going to talk about the 3 simplest, which are kernel depth, kernel height, and kernel width. So pay attention to those measurements and what they mean, because they're going to come into play when thinking about how the kernel grows on the cob. And so the data that I'm going to talk about today come from the running the nested association mapping population, which I'm sure you guys are all very familiar with. So what we did was we sampled 96 seeds from each of these parental lines, and to survey the potential phenotypic variance for our machine vision pipeline. And so by doing that, we have collected over measurements for over 2,300, paired measurements for over 2,300 kernels. And so just to get a sense of what we've accumulated here, we have 2,300 measurements of NIR data, which creates an NIR data matrix, which is 2,300 rows and by 790 columns, right? A very large matrix of information. And then we also have this shape data matrix, which we're collecting from the images. And so what we're going to attempt to do is try to find relationships that exist between the NIR data and then the shape data. The way we're going to do this is by using a method called canonical correlations analysis. It's probably not something that you're very familiar with, but you just need to know that it's similar to principal components analysis, which I think most of you are familiar with. And it seeks to find linear vectors through two data matrices that have maximum correlation. And so this, in effect, is a latent parameter model. So we're finding latent parameters that are correlated between the two matrices. And each trait has a weighting on each canonical variate. So the linear vectors that we're pulling from each data matrices are called canonical variates. And so I'm going to walk you through one of these analyses briefly. So what we're going to do is we're going to take the NIR spectrum and the imaging spectrum, and we're going to run it through CCA, and canonical correlations analysis is going to pull out the first canonical variate. That first canonical variate is going to be some form of the NIR data and some form of the morphology data. And those two canonical variates are going to have the maximum correlation that could possibly exist between these two data sets. We're going to repeat that and find the second canonical variate, and so on until you pull canonical variates that no longer have any significant correlation with each other. So for these two data sets, we found that there are three significant relationships. And the way to read these data are the first canonical covariate variates have a correlation coefficient of 0.82, which is quite high, and that explains so the way this is a redundancy index and allows you to say how much of the variation in the kernel morphology data is explained by the NIR data. And the first canonical variate explains 19% of the total phenotypic variation in the kernel morphology data. If you add all three significant canonical variates up, 44% of the phenotypic variation in the seed size data is explained by the NIR, which is quite a bit. But for time's sake, I'm only going to talk about canonical variate 2. So the way to read this graph is the canonical variate scores, which are the new traits that you are producing, don't really have any biological meaning to us, right? What does a canonical variate score of 1.2 mean? It doesn't really mean anything. So what you have to do is you have to rotate that back into a biological framework. And you do that by doing a correlation between the canonical variate 2 score and the original variable. And so that's shown here. So for example, the canonical variate 2 score is positively correlated with the height and the width of the kernel and negatively correlated with the depth. And so if you look at the three different metrics, what that means is the canonical variate 2 is positively correlated with the cap area. So as the cap area gets bigger, the canonical variate 2 gets bigger, but negatively correlated with depth. And so what that means is as the kernel gets fatter, it gets shorter, right? And so canonical variate 2 is basically telling us how the kernel grows on the ear. On the flip side, instead of doing a correlation between canonical variate 2 scores and the NIR data, which isn't going to mean anything to anybody, what we do is we do the correlation between the traits that we can pull from the NIR. And these traits are weight, volume, density, oil, the things I showed you before, right? And you can see that what is related with canonical variate 2 is primarily weight, volume, and density in a positive correlation, and oil with a negative correlation. So what that means is as the kernel gets shorter and fatter, the weight goes up. Right? So the weight is positively increased with a large cap area. And so what we can do is we can then sort based on canonical variate 2 the images of each one of our kernels. And you can see it very clearly here where you have a tall, small capped kernel with a low canonical variate 2 score. And as you move along canonical variate 2, you end up with a short, fat kernel with a large cap area, right? And so canonical variate 2 seems to indicate that large round seeds have increased density and protein but decreased oil. Now when I saw this, if any of you ever had an ear of corn in your hands, the first thing I thought of was as you look across that ear, you have regions of the ear where some kernels are packed very closely together and they're tall and thin, versus other areas of the ear which don't have such tight kernel packing, and which what ends up happening is that the kernel fills the space and it's short and fat, right? So my initial impression with this, it's telling us something about kernel packing on the ear. And so what I've showed you here is an ear of B73 that shows that very phenomenon, where you have some kernels that are tightly packed together, right? They have neighbors that are very close together. And as you move along towards the butt or the tip of the ear, you have these guys that are now a little shorter, rounder, fatter, because they have a little more space to grow. And so as a sub-experiment of this project, what we did was we created ear maps of all of the NAM parents. And what that means is that because we can do single kernel measurements, we can then take each one of these kernels, assign it a position on the cob, and then run it through our pipeline. And what we can do is then relate it back to its position on the ear because we have that information stored. And so because we have all that information, we can then calculate a canonical variate 2 score for each one of the seeds as it lies along the ear. And when we do that, we see that a low canonical variate 2 scores for these guys right in the middle, right? These very tightly packed, close neighbor individuals. Whereas as a very high canonical variate 2 score are these individuals over here which are fat and round. So it really looks like canonical variate 2 is telling us something about how kernels pack on the ear. Of course, there's also genotypic differences. Some genotypes have widely spaced kernels that are fat and round, right? But I think canonical variate 2 score could reflect that also. But I think this is very enticing. So I wanted to ask one more question with this. So what I did was I took an ear from just from a bin somewhere of a hybrid, an inbred line that was not included in the model, W64A. And it's very clear that this has seed set issues, right, on one half versus the other. And so to test this theory, what I did was I divided the ear in half and measured sent the kernels through the NIR machine and measured a canonical variate 2 score for each one of the halves. And it's very clear that the distal half, which is much more tightly packed than the proximal half, has a much lower canonical variate 2 score, indeed confirming that it looks like that canonical variate 2, if the score itself doesn't mean much, but if you rotate it back into a biological context, it looks like it's telling us about how kernels grow and develop and the kernel spacing on the ear, which is very interesting. Okay, so I would like to then move on to our measurements of root growth. And so what I just described was, I think, using the NIR to kind of go backwards in time, to talk about how the NIR can tell us something about how the kernel developed as it was growing on the ear. Right? And I think that's very interesting. But we can also go forward in time, but to try to estimate how, does the NIR say something, does the profile of this NIR profile of the seed say something about how that seed's going to grow when you put it in the ground? And so the protocol that we're going to go through, so each seed obviously is an indexed order, so it gets imbibed in indexed order in these microtiter plates in heated water. So once the seeds start germinating, they get placed on an agar slab where they are, and then sandwiched in between 2 pieces of agar. They get then placed in front of fixed position CCD cameras illuminated with NIR. And after a short period of acclimation, the plate is rotated so that it induces rotated 90 degrees to induce a gravitropic stimulation. And then we measure the growth of the root every 3 minutes over 3-hour time course. And what you end up with is something that looks like this. So this is one plate, and the colored lines are just extended from the tip to highlight the movement of the roots. And if this is going to work, yeah, there it goes. So that's over 3 hours of growth, a root responding to the gravitropic stimulus. And what this produces then is a movie of root growth, from which you can extract any number of phenotypic measurements. And so what I'm showing here is extraction of tip angle over time. And so if you look at, these are 5 different genotypes with different response. This is the average tip angle response of 5 different genotypes over time. And you can see they're quite variable. And so what's important to know here is that once these movies are collected, you can go back and apply any kind of algorithm to extract as many phenotypes as you like. We've also extracted, one important one is growth rate, how fast the roots are growing, as another example. But primarily today I'm going to be talking about tip angle. Again, we generate this data set where we have the NIR matrix on one side, and now we have the root growth rate data on the other. So before, when I was talking about the shape data, we were using about 2,300 individuals. So we recently got data paired data for over 7,000 individual kernels, where we have both the NIR data and the root growth data. And this represents over 600 genotypes, NAM parents, Wisconsin Diversity Panel, B73, NC350 RILs, and then classic composition mutants. So a wide diversity of traits. And what I'm going to show you is probably the most simplistic way of looking at this data. It's simply asking the correlation between the traits that we pull from the NIR and the tip angle. So for example, this is the correlation between weight over tip angle time, right? And what you can see is that the correlation, and the dotted line on here is a Bonferroni adjusted p-value of 0.01. So anything that dips below or above this line is highly significant. And you can see that weight is having a significant influence on how the kernel weight is having a significant influence on how the root behaves. What's interesting is that density also seems to have a significant influence, but only at the very final time points which you're measuring is the final tip angle, the resting tip angle where we end the measurement. And so what this looks like it means is that kernels that are bigger have a higher final tip angle, but the kernels that are a lower density also have a higher tip angle. So high density kernels don't swing as far, which is interesting. And lastly, oil is significantly correlated, but at a different time point. So oil seems to be correlated at a time point when the root is just adjusting to gravity. So we call this swing rate. So it's how fast the root adjusts back to the gravity vector. And oil seems to be having a positive influence on that. I think that may have something to do with embryo size relative to the kernel. What I want to say here is that if you notice the y-axis, these correlation coefficients are very low, right? They're highly significant, but they're very low. So what that means is that I think it says something about the power of our method to be able to detect very, very small significant relationships. But I think it's important to be realistic here in that we're using an NIR profile to try to estimate something that happens later on in life when the root is growing. And I think it's really impressive that we're able to find these significant associations between traits, kernel traits, and growth traits. So this is one way to look at the data. Another way is to say, well, okay, if there is a relationship between NIR and the way the seedling grows, well, we should be able to predict the growth profile from the NIR spectra, right? We have a very low relationship, but that relationship does exist, and so therefore we should be able to do some sort of prediction about the way the root is responding when it grows. And so again, we're gonna do our calibration, and instead now we're gonna switch out the analytical data for the gravitropic profile, right? And then we're going to take the calibration model that we've created. We're gonna put in a novel NIR spectrum and then predict a gravitropic profile for a particular seed. And then since we have over 6,000 trials, we have a very large data set to then validate this prediction. So we're going to take the predicted gravitropic profile compared to the actual gravitropic profile, and then to do the validation. And that's what this figure shows. So this is going to be the correlation between the predicted gravitropic profile and the actual gravitropic profile. And what you can see is that the correlation maximizes out here at the final tip angle, but it's also significant back here when the root is just swinging back towards gravity. And the significance level here is established by a permutation threshold. So you randomize the data to establish the correlation coefficient of randomized data. So I think this is really significant that we're actually using the NIR to be able to say something about how a root's going to respond when you put it in the ground. But what can we do with this? So what's the utility of this? One way of attacking these highly significant yet low correlated values is to be able to enrich for a favorable outcome. So I think it's unrealistic to expect that we can take an NIR profile and completely predict the gravitropic profile, right? I think that's really unreasonable to expect. But what we might be able to do is to take the NIR profile and to enrich for an outcome that you desire. So let's say, for example, that you want to get rid of the low responders. In this case, the low responder is going to be something that has a low tip angle, right? It doesn't respond very well to gravitropism. And if we want to get rid of those guys, if we separate the data set into a high responders, mid responders, and low responders, we can then ask the NIR to predict whether you're going to be a high responder, a low responder, or a mid responder. And this table here is what's aptly called a confusion matrix, or because it's difficult to interpret. But what we're saying here is that if the NIR has predicted it to be a high responder, 33% of the time it actually is a high responder, right? Likewise, if it's predicted to be a high responder, 48% of the time it's a middle responder. And only 17% of the time when we predict it to be a high responder is a low responder. So if you choose those high responding individuals, you're going to enrich by 83%. So if you take the summation of the middle and the high responders, you get 83% of your kernels are going to either be a middle responder or a high responder, therefore getting rid of the low responsive class. And so if you were to just take it random, those numbers will be, you would estimate that 66% of your, because we're separating them into 3 groups, 66% of your data would fall somewhere in the middle or high class. If you run the kernels first through the NIR and then predict its response, you can get that number up to 83%, which is an enrichment, of almost 20% over what you would expect by random chance. I think it's a very reasonable expectation that you're able to do this with NIR. Instead of predicting the entire response, we can try to enrich for favorable outcomes. Now, we may not be interested in trying to predict gravitropic response, but you might envision that this is something you could use this for germination rate under cold conditions or some other phenotypic response. Okay, so I think I've told you that the machine vision pipeline is used for identifying novel seed and seedling relationships. I've shown you relationships exist between kernel composition and shape, and that CV2 seems to be a measure of packing on the ear, and that there's a negative relationship between seed packing and weight and density. And I also showed you that kernel composition, there is a relationship between the kernel composition and how that seed is going to behave when you put it in the ground. There's a positive relationship between swing rate and relative oil content, which is interesting. And I think the utility of this, extrapolating NIR to more extant regions of its growth profile, is that you can exploit those relationships to enhance for desired outcomes, such as increasing root responders, as I showed you. So with that, I would like to acknowledge the PIs of the lab are here, highlighted here in bold, Mark from my lab. John Byer from our lab is the technician, as I showed you, who does he feeds the machine, and he does a lot of that. Edgar is the PI in Wisconsin, and Nathan Miller is the brains behind all of the algorithms that are written to extract the data from the tip angles and the shape. And so he's a real asset for us. Paul Armstrong built the NIR machine, and then Teresa is a professor at an undergrad college that has done some other work. And I want to also mention that Takeshi is the guy that puts all of the germinating seedlings on the auger and watches them grow over time, which is a lot of work. And Bessie Split does our imaging of the seeds. So with that, I'll take any questions.